Hi, I'm Victoria, and I'm going to be going over prompt number 1. Here is a diagram of a nucleotide, which is the basic building block of DNA. They contain a phosphate group, a 5-carbon sugar called deoxyribose, and a nitrogenous base. Let's take a closer look at deoxyribose. Our 1' carbon is where the nitrogenous base forms a covalent bond. The 2' carbon is missing an oxygen compared to ribose. The missing oxygen distinguishes this molecule is the deoxyribose found in DNA. The 3' carbon joins to the 5' carbon of the next nucleotide. The 5' carbon forms a covalent bond with our phosphate group. Notice how our phosphate group has a negative charge, giving DNA its negative charge. The nitrogenous bases in DNA include adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, which have a double ring structure. Cytosine and thymine are pyrimidines and have a single ring structure. As you can see, G always pairs with C and A always pairs with T. This makes our 2 DNA strands complementary to one another. When the nitrogenous bases pair, they form hydrogen bonds. A and T form 2 hydrogen bonds, while G and C form 3 hydrogen bonds. These hydrogen bonds are relatively weak. Think about taping something together. It holds things together to an extent. The strands are also anti-parallel. Notice that the 5' carbon of one strand lines up with the 3' carbon on the other strand. Base stacking in DNA is where each base pair is parallel to the other. This helps stabilize our DNA. The picture on the left shows a left-handed spiral, and the picture on the right shows a right-handed spiral. DNA is most commonly shown as a right-handed spiral with 10 base pairs per turn. DNA also has major and minor grooves. As you can see, the major groove has much more room for protein binding and interaction. If we place our DNA in a concentrated saline sodium solution, it will help stabilize our DNA molecule. DNA has a negative charge, which causes the strands to slightly repel each other, and it is destabilizing. Sodium has a positive charge, which helps stabilize the DNA. Since the DNA is more stable in the sodium solution, it will have a higher midpoint of thermal denaturation, which is a temperature where 50% of the DNA is denatured. This is because the DNA strand will be more stable.